like a lamp? Yeah, wait, hold up.
Ah!
Hello and welcome to our meiosis video. First, let's go over some definitions. A chromosome is a thread-like strand found in the nucleus, and it contains DNA. Here we have a single strand, chromosome I, and a double strand, chromosome X, which contains twice the genetic material. Next are homologous pairs. Two chromosomes, one from mom and one from dad, that carry the same genes and are similar in shape and information. Next up are centromeres. The centromere is the center point of a chromosome and can be thought of as a connection point. A diploid cell contains two sets of chromosomes, one from mom and one from dad. A 2n = 6 cell will have 6 total chromosomes and 3 from each parent.
The nuclear envelope contains the nucleus, which is all chromatin. Chromatin is uncondensed DNA. During interphase, the chromatin replicates and condenses into chromosomes. The centrosome also replicates. The centrosome is the source for microtubules, which are not yet apparent in interphase. We will discuss this more later.
Here we see the chromatin having condensed into chromosomes. The centrosomes began to move apart, which makes the microtubules apparent. Microtubules later act as a track for the chromosomes to travel along. The nuclear envelope also begins to break down. Once the chromatin has condensed, the chromosomes line up with their homologous pairs and begin the process of crossing over. The crossing over mixes the mom and dad chromosomes results in new mixes of genes as opposed to them being separated by parent.
Chromosomes line up along the metaphase plate. The centrosomes are on opposite ends of the cell and have microtubules connecting them. The chromosomes are now connected to the individual microtubules at the centromeres.
The homologous pairs begin moving apart along the microtubules. The cytoplasm, which is a jelly-like substance that is in the empty space in the cell, begins to split to form the two separate cells. Each cell has 3 chromosomes, and a nuclear envelope forms around the nucleus in fragments. There are 2 haploid cells. Each will undergo the process of meiosis II individually.
The centrosome replicates once more. The centrosomes begin to move apart and the microtubules are apparent. The double-stranded chromosomes line up.
The centrosomes are at opposite ends of the cell again, with microtubules stretching across the cell. The double-stranded chromosomes line up with their centromeres along the metaphase plate. The double-stranded chromosomes separate at the centromeres and begin to move along the microtubules. The cytoplasm undergoes cytokinesis. The chromosomes begin to turn back into chromatin, which also forms the nucleus. There are now 4 haploid cells.
We started with a diploid cell, which had 6 chromosomes. Those replicated, which led to a total of 12 chromatids, or 6 chromosomes. Through meiosis, 4 daughter cells were formed, each with 3 chromosomes. Hey, that is also 12. These daughter cells are unique from one another, and the original cell. They are sex cells, such as sperm and eggs. Another word for a sex cell is a gamete.
I know that these are little.
Oh god, I forgot. Bleep.
Bleep, yeah, no cursing. Not like David dropping the F-bomb again.
That
It was just uncalled for.
All right, you ready,
You
homie?
having...
Oh,
Okay,
wait,
you
it's
have
this
to
window.
Okay, are you guys ready? Stop. And then you pull up closer to the thing. So go. Wish me luck, everyone.
Good luck.
This is some high-profile action. Oh, she running. All right, that was.
That was the adventure.
Wait, I can't see you. Here you go.
What do you mean you can't see?
You couldn't
Back up!
hold the camera.
Back up!
All right, so this is here.
Signing off. Oh, whoa. Signing off.
Hey, it's David.
Bye.
Bye.